Bringing a new cancer drug to market is an exhausting marathon. It requires up to 15 years of dedicated research, navigating dense regulatory hurdles, and spending billions of dollars in development. Even with that massive investment, the failure rate is exceptionally high. Most candidates are abandoned, and the few successful chemotherapy drugs often leave patients struggling with severe toxicity and side effects. To bypass these early safety hurdles, scientists use a strategy called drug repurposing. They take a medication already approved for one condition and test it against an entirely different disease. Finding a cancer treatment inside a decades-old medication bypasses the typical development pipeline entirely. If a cheap, pre-existing pill can treat a complex malignancy, it upends an entire pharmaceutical model built on exclusive patents and high-margin therapies. Glioblastoma is an aggressive, notoriously fast-moving brain cancer. Average survival times are short, and the disease is widely considered incurable. A few years ago, researchers at Johns Hopkins were setting up a routine animal study on the disease. They were preparing to implant human glioblastoma cells into a colony of laboratory mice. Right before the study began, the lab encountered a standard issue, a pinworm infection in the colony. To clear it out, the mice were fed Fenbendazole, an incredibly common, dirt-cheap veterinary dewormer used worldwide for over 60 years. This published report outlines what happened next. When the researchers finally implanted the glioblastoma cells into the mice treated with Fenbendazole, the brain cancer failed to develop. It never grew. A basic animal antiparasitic, designed to clear intestinal worms, had completely blocked the engraftment of a lethal tumor. Scientists immediately needed to know how. To understand the mechanism, researchers moved from the animal lab to the petri dish. They isolated human cancer cells, including lung, breast, and colon lines, and directly exposed them to Fenbendazole. To rapidly divide and multiply, a cancer cell relies on a biological highway system made of protein structures called microtubules. If you break the highway, the cell cannot split. The Fenbendazole chemically binds to the tubulin proteins within those highways. This destabilizes the microtubules, paralyzing the cancer cell and locking it in the middle of its division cycle. But halting division isn't enough to destroy the tumor. Paralyzing the cell is simply the opening move in a highly coordinated biological attack. Cancer cells are metabolically demanding. They are heavily addicted to glucose, consuming massive amounts of sugar to fuel their rapid growth. This diagram maps the cell's metabolic pathways. At the bottom left, Fenbendazole interferes by downregulating the enzyme HK2, starving the cancer cell of glucose energy. Shifting to the top, this starvation pushes the paralyzed cell into an inflammatory self-destruction sequence known as pyroptosis. During pyroptosis, the cancer cell rapidly swells. Microscopic pores form across its membrane, and eventually the cell violently bursts, expelling its contents. By combining structural paralysis with metabolic starvation and an explosive inflammatory trigger, Fenbendazole systematically dismantles the cancer cell from the inside out. Microscopic petri dish reactions are fascinating, but scientists need to prove the drug functions the same way inside a living organism. These photographs display extracted tumors from a live mouse model battling breast cancer. As the dosage of Fenbendazole increased, the physical size and weight of the tumors shrank significantly, and as these charts indicate, the mice did not experience the toxic fallout associated with traditional chemotherapy. They maintained steady body weights, and tissue slides showed virtually no damage to healthy organs like the liver and kidneys. Fenbendazole cleared the most difficult hurdle in early oncology research. It demonstrated the ability to aggressively target the tumor without killing the host. Today, Fenbendazole remains a generic, off-patent veterinary drug. You can find it sitting on the shelf at local agricultural supply stores. While social media is filled with viral stories of patients claiming remission after taking the dewormer, the medical community requires definitive proof. Right now, there are no large-scale, controlled clinical trials proving its efficacy in humans. Moving the drug to human trials presents severe biochemical hurdles. Fenbendazole has extremely poor water solubility, which means predicting how well the human body will actually absorb and distribute the chemical is incredibly difficult. Isolated laboratory successes and internet testimonials provide interesting data points, but they cannot replace the rigorous clinical monitoring required to guarantee patient safety and exact dosing. The final barrier is purely financial. Running the multi-phase human clinical trials required by regulatory agencies costs hundreds of millions of dollars. Because fenbendazole's original patent expired in the 1990s, no pharmaceutical company can secure exclusive sales rights. Without a patent, there is zero financial incentive to fund those massive trials. Medical professionals maintain a strict boundary. Attempting to treat complex malignancies with unapproved veterinary drugs outside of clinical supervision carries immense risk. The story of fenbendazole highlights a frustrating reality in modern medicine. A highly effective biological mechanism against cancer exists on store shelves today, stalled not by a lack of scientific promise, but by the rigid economics of pharmaceutical companies.